Heydari, and it is ACTIVA realistic single cell generation with automatic cell type identification using introspective variational autoencoders. Ali, if you can take it away.
Hi, can everyone hear me okay?
Yes, we do.
Awesome, thank you so much. Yeah, good morning. It's my absolute pleasure being here. I'm sharing with you some of the work that we did on generating realistic synthetic RNA single cell RNA sequencing data. And yeah, I just hope where you're at is not as early as here in California. It's around 6 a.m. So if I yeah, so let's just get started. So I was gonna introduce like why we have single cell resolution, but I think Chisun did a phenomenal job of explaining it, and I also have the smoothie analogy, so maybe I can skip over this part. But yeah, the idea is that with single cell RNA sequencing technology, we can get the gene expression at a single cell level, and then from that we can delineate heterogeneity that we might have. So in a lot of cases, for example, in cancer, if we're trying to look at a tumor, there's a lot of heterogeneity. There are different cell populations that are present. So it's really useful to know individual cell populations that we have. So that's the very brief overview of why we need the single cell technology. But the question is, why do we need to generate synthetic data? And why do we need to have this option to augment existing data sets? So as you all may know, single cell RNA sequencing is really expensive and time consuming. And it's also an invasive procedure. So it might be, so a lot of times when we're trying to obtain samples from the mice studies or other animals, we have to sacrifice them. So that's another disadvantage. Also, sometimes it's just hard to obtain the single cell RNA sequencing. So if we had an option to augment the existing data sets or to generate new ones if we needed it for, say, doing trying to see if our machine learning model or benchmarking study, how it works, so that would be really cool to have an option where we can generate realistic single cell data. And when we were talking about generating realistic single cell data, it's what you would expect in like a very broad sense. So if we want to think about it in computer vision, this has been done with GANs and VAEs, where we feed in a noise vector to a generator model, and this generator model can produce data that's similar to the initial to the training data, but it's new, it's different. So instead of doing this with faces, we want to do this with single cell RNA sequencing data. So this is what we mean when we say we want to generate synthetic data. And the data that we work with, it's the gene expression data. So we have cells by genes, and the order of so the number of genes that we have is often on the order of thousands, so 20,000, 17,000. And an issue here with doing some of the more traditional machine learning studies is if we have the same number of features as we have in the same number of samples, which is often the case with single cell RNA sequencing. A lot of the traditional fundamental machine learning algorithms aren't able to learn very well. So that's why we want to use deep learning model to do so. Now, at this point, you might be asking, well, have people done this with traditional statistical model? And the answer is yes. But as it was shown by another deep learning model, SCGAN, which is our competitor, and that's what we're going to compare our model with. The statistical model that have been done in this area aren't nearly as advanced, and they don't produce as much of a realistic data as the deep learning models. So that's why we want to do a deep learning model, and then we're going to compare what we were doing with the existing state of the art, which is SCGAN and CSCGAN by Maruf et al. So the question for our model design is we have 3 points. One, can we design a model that can learn the underlying manifold of the data? Two, can we do this while we're learning individual cell populations, and specifically the rare cell populations? And lastly, can we do this effectively and efficiently, where we can generate these cells on demand very quickly, and we can also train it very quickly and in a stable manner? So because of the time, I'm going to skip over a lot of the details of the algorithm design, but we do have a preprint available, and I'll be happy to answer any questions as well. So I'll be giving a very brief overview of how we're doing so. So we want to use a variational autoencoder to do this, and the reason for it is because variational autoencoders are very elegant, and they're theoretically pleasing. There's a lot of cool math that's behind it, and training them is very stable. In addition to that, it's also easy to implement them. So that's why we want to use a variational autoencoder as the base of our model instead of, say, a GAN-based model. But there are two main issues with using variational autoencoders. The first issue isn't as pronounced as the second one, but there's this issue of posterior collapse, which I'll talk about in a second, what it is and how we can potentially prevent it. And the second one, which is I think the main issue, both in computer vision and in other areas, is the fact that vanilla VAEs produce blurry samples. So they adhere, so the samples that they produce adhere to an average sense in the data. So in this, and with our framework, we wanted to see how we can alleviate these two issues and see if once we did that, if we were able to generate data that's as comparable to GAN-based model or even better. So with the first issue with the posterior collapse, that's when there's a lot of theoretical work that's been done in this area. But just to broadly summarize what it means, that's when the generator. So posterior collapse happens when the generator decides to ignore a subset of the latent variables. So that means that the samples that you're producing aren't as diverse anymore. So here I have an example of a semi-collapsed posterior. So as you can see, all the faces are pretty similar, but this issue has been linked to over minimizing specific parts in the loss component. So the work that we did and some other folks in the field have done is to show that if we avoid this over minimization of individual loss component, we might be able to alleviate this posterior collapse. So this is using our algorithm that we have called soft adapt. If we do that, we can actually avoid this from happening. So that's our first approach that we're trying to do to avoid the first issue with VAEs. But the second one, which is a little bit more important, and it's kind of the meat of our method, is how can we generate samples that aren't blurry anymore. So here I'm showing an example of generating like faces with vanilla VAEs. And if you compare it to like the GAN-based model, you can see there's a clear difference. But there was a work that was done in this area. And these models are called introspective variational autoencoders. And introspective variational autoencoders kind of mimic what GANs do, but they do it in a very efficient way, and they kind of recycle the existing components. So we'll get into that next. But before getting into the introspective variational autoencoder, let me give a brief overview of what GANs do and why is it that GANs produce such high quality samples. So as most of you are probably familiar with, GANs have two networks. There's a generator network and there's a discriminator network. The generator network tries to generate new data from this noise vector. And there's a discriminator that decides whether the samples that are being inputted, if it's real or if it's fake. So in the beginning, you have real data coming in. That's very different from the generated data because the generator is just generating nonsense. So in the beginning, it's really easy to distinguish the difference between the two. And based off of that decision, both networks are being, there's back propagation and both networks are being trained based on that feedback. But as the generator keeps getting better and better, the discriminator is having a hard time to decide between the two. And based on that, there's this min-max game that's being played. And that's what allows GANs to produce really good samples, realistic samples. So we want to mimic this idea for a variational autoencoder. And the idea is that what if we just recycle the encoder that we have in a variational autoencoder, and we use that as a discriminator. And the idea behind that is that, well, if the encoder is doing a really good job of learning how to map the real cells to this encoded space, then the generator must learn how to map these lane vectors back to the original space. And if we feed that back to the encoder, the representation, this ZR here from the reconstructed cells and the original representation that the encoder had should be very similar. So that's kind of like the broad idea of why we want to recycle this encoder as a discriminator. And this is where like the first part of the adversarial loss comes in. So we're looking at, so these two components, this LREG, which is the negative of the KL divergence, and this LAE, which is the autoencoder loss, so the MSE. These are just what you would expect to see in a vanilla VAE model. But this additional component here is what it's the first part of this adversarial loss that we're defining. So this M is an adversarial constant, and this KL divergence is being done on the samples that the generator is producing. So as you can see, the way to minimize this is to for the encoder is to maximize this value to match M. So that's the first part of the adversarial training. And for the second part, so this was again the loss component for the encoder. For the second part, we're trying to minimize this these generated samples that the generator, so we're trying to minimize this regularization term of the generated samples. And that's how the generator is trying to minimize it while the encoder is trying to maximize it. So that's where the adversarial training happens between these two. And one thing to keep in mind is that the generator is actually generating two things. One is going to be the reconstructed cells, where we feed in real cells here, we get a latent vector, and then we feed it back to the generator. So we call these the reconstructed cells. And then the second one is what we call the generated cells. So these are the brand new cells that we're going to generate out of a noise vector. So at the same time, we can think about it as like a second pass. We're passing in these noise vector to the generator. And the idea here is that if the generator is doing a really good job of learning how to map back these latent vectors to the original space, if we feed it a noise vector, it should also be able to do the same thing. And that way we can compare this encoded generated cell with the real cell that the encoder had initially mapped to that latent space. So that was how to essentially fix the blurriness of the VAEs. The second part is how can we avoid the posterior collapse. And as I mentioned earlier, we're gonna use this algorithm called soft adapt. And what soft adapt does, it looks at individual components of the loss, and it looks at how fast and how much they're decreasing. So if one loss component is decreasing really fast, really quickly by a large value, then it's going to put a stop on it and make sure that the other loss components are being trained. With that said, even though we did this, we didn't really see posterior collapse happen within our training. So we think that even if you use fixed weights, you should still be able to get pretty good results. But in case if you happen to run into posterior collapse, then this is the framework that, so this is already embedded within our framework. Now, the second part, this is, I think, also like a really novel thing that we want to do was to learn how to make sure the model is generating cells from all cell populations and not just the one with the majority of the cells. And for that, this was our inspiration, because a lot of times when we're generating data, synthetic data, the minority populations or even some populations that aren't just as pronounced in the original data, they're being disappeared. So here we're showing that example within these two population where like there's more, obviously there's a lot more in the real data, but the GAN-based model is like failing to produce a good enough amount of it. So for that, we wanted to introduce this cell type conditioner. And the idea is that we want to see all the cell populations that are within the real data. And as we're reconstructing the cells and as we're generating cells, we want to make sure that these cells that we're generating have the same number of populations as the real data. So this is going to allow us to, one, learn the cell types. So if we want to do classification, we can do it all in one go. Two, we can also do this in an efficient manner where we're also learning the real cell types. And if we're trying to produce specific cell populations, which is the goal for doing data augmentation, we can do that with this framework. So even though in our framework, this is very broad, so you can use any classifier, we specifically use Actin, which is a state-of-the-art neural network for doing the cell classification. So now putting everything together, we do this all. So these are our loss components that we have. And as I mentioned, the alphas are the adaptive weights or fixed weights that we want. And M is this adversarial constant that we find out as we do like the warm-up beforehand. And if we want to put everything together, this is how it looks like. So it looks kind of complicated, but this is actually all in a single stream. So that's why, so that's where the efficiency comes in. So first, this is kind of like the reconstruction part, where we're reconstructing all the cells. And then secondly, we're learning how to generate brand new cells that we haven't seen before. So that's the second component. So, and as I mentioned, ACTIVA is single stream. So this happens all very quickly, much quicker than like a GAN-based model with the scheduling that happens. So the two models that we're going to compare now, I'm going to talk about our results. So the two models that we're going to compare against are the two state-of-the-art models, the CGAN, which is generating data from all populations, and the conditional CGAN, which is generating specific cell types. And we have one model that's trying to compete with both of these models. So that's also another thing to keep in mind. So if you want to have both of these capabilities, you have to train two different models. With ACTIVA, you can train one model that can do both of these. And the data sets that we're going to use are fairly standard, the 68K PVMC and the 20K brain small. And here are the results. So the first thing we want to do, we want to see if we could qualitatively make sure that the data that we're generating looks like the real data. So here we can see that the data that we're generating looks like the real data, but also we have more cells per population than the real data. So that was another goal, because we want to make sure that we have all cell types and specifically the rare cell population. So we can see that qualitatively, we can see that here, and also the gene expressions match. So that's a really good sign. But quantitatively, how can we measure how well we're generating data? So one way to think about it is that, well, can we distinguish the data that we're generating against the real data? If the answer is yes, then that's not a good thing. We want to make sure that a classifier has a really hard time distinguishing between the data that we're generating and then the real data. So in this case, it's kind of counterintuitive, but we want the AUC for this classification problem to be closer to 0.5, because we want the classifier to be confused between distinguishing our data and the real data. So for both data sets, we can see that ACTIVA is doing a much better job than SCGAN, which is the current state-of-the-art model. And we can also do this for a specific cell population. So what if we generate a specific cell population? We can ask the same question. Can a classifier distinguish the difference between our data that we're generating and the real data? And in that case, also, we see that ACTIVA is doing a better job than CSCGAN, which is the model that generates specific cell populations. So these were really good. These were really good signs of our model doing a good job. But I think the kind of like, the cherry on top is this problem. So this is how we think that ACTIVA can be used. And the idea here is that, can we augment existing data sets to make them better for classification or other problems? So can we take in rare cell populations, learn with ACTIVA how to generate more of it, and add it back to the real data and see how that works? So we do this experiment where we progressively decrease the number of samples that we are using to train ACTIVA for a specific cell population. And then we put back those. So once we train ACTIVA, we generate the cells from that population that we downsampled, and then we feed it back to the, we put it back to the data. So we augment the original data, and then we do a classification to see how well we classify. And here we can see that even if we train the data on about 0.5% of the of the total number of cells that we had initially, if we augment the data with ACTIVA, we can get a really high accuracy. So this is really promising, because that's telling us that if this happens in a real case scenario where we have a really rare cell population, training with ACTIVA and augmenting the original data with ACTIVA can improve the downstream analyses that biologists want to do. And here, if we do it quantitatively or qualitatively, we can see that the space where ACTIVA is generating these cells is very similar to the real space that these cells were originally embedded in. And finally, we talked about efficiency. Well, how efficient is ACTIVA? We can see that not only ACTIVA can produce better quality of cells compared to like SCGAN and CSCGAN, but it can also do it in much faster time. So these are the training times under the same computational environment. And we can see that like here for 68K, we do this in 7 hours, while SCGAN does it in 45 hours and CSCGAN does it in 48 hours. And one other thing to keep in mind is that if you're trying to do both of those things, you have to train 2 separate models, while with ACTIVA, you can train 1 model and you kind of have both capabilities all in one. And to kind of summarize what I talked about today, we introduced the novel deep learning model for generating realistic synthetic single-cell RNA sequencing data. And we had two goals in mind. One, learning all cell populations and generating rare cell types, and 2, doing that in a very fast and efficient manner. And we showed that if we use ACTIVA to augment a very rare cell population within our data, we can in our experiment, we saw improvements more than 45% when we're trying to classify it. So I gave a very brief overview of everything. We do have a preprint that's up online. So please take a look at it, and I'll be happy to answer any questions you might have. And just to acknowledge our collaborators, these are the folks that worked on this project. Thank you so much.
Thanks, Ali. It was a great talk. We have almost like a time for one question and end of the session. I don't have any question from the audience. So I have one question, probably quite irrelevant to the whole mathematical description. So what I was wondering is what happens in the downstream analysis? Like, think about the trajectory when you're looking at the cell development. So adding these, basically learning from the noise and the cell, adding these new cell types may not biologically simulate those cells. So have you ever tried to look at the downstream of some of these rare cell types you're adding and how it's affecting the downstream analysis?
Yeah, that's a great question. So we did, I would say we did 3 studies that we tried to look at. One was the classification that I presented here. We wanted to see if we can do a better job of classifying that rare cell population if we add the augmented data. So I presented those results here. The other thing that I think we looked at was to see if we add these, if we add this augmented data, what are the important features within, like, can we pick up the important features that we would have had in the real data and the augmented data? And we saw that there is, so I think I have those results here. So, sorry, so here we can see that, like, if we do this with, sorry, I think my PowerPoint just went on. So there we go, okay. So here, sorry, if we add this data basically back to the, like if we augment the data of the features that we get, the important features that we get, a lot of those features do show up. So we think that, as you mentioned, so there will be some noise that we're adding, but we think that because we're learning from the data in a very broad sense, we are picking up those important features from the rare cell population. So if we add it back to if we augment the data with this new information, we think that the top features are going to show up again. So those are the very, I would say, preliminary analysis that we've done on the downstream, on the downstream analyses.
Great, thank you. With that, we will going to conclude the first session. We're going to have a very short break before our keynote speaker. So I think the time back, we're going to have about 10 minutes break, and the next session start 9:45 Eastern time and 15, if I'm not mistaken, 15:30 or 15:45 Central European summertime.
Yes, thank you. I will share another postcard from Padova as yesterday, just to keep you company until the keynote at 3:45 p.m. Central European summertime.